Today we are going to be talking about cell signaling, and we are going to be featuring the special guest G proteins. So just to get us started, here's a super basic overview of cell signaling. So here we've got step 1, which is reception, and reception is the target cell's detection of a signaling molecule coming from the outside of the cell, because this right here is the plasma membrane. And then the second step is going to be transduction, which is what is happening right here, which is the binding of the signaling molecule changes the receptor protein in some way, which initiates the process of transduction. The transduction stage converts the signal to form that can bring about a specific cellular response, and that is where the third step takes place, which is the activation of cellular response. And that again is a super basic overview of cellular signaling, and now we're going to get into more specifics with the G protein. So just a little bit of background before we get super into it. A G protein coupled receptor is a plasma membrane receptor that works with the help of a G protein, a protein that binds the energy-rich molecule GTP. And so now we're going to get super into the steps, and then we're going to see its applications as well. So here we've got the first stage. And so we have the loosely attached to the cytoplasmic side of the membrane. The G protein functions as a molecular switch that is either on or off depending on which of the two guanine nucleotides is attached. And so here we have GDP currently attached, but right now the G protein is inactive. And so the receptor and the G protein work together with another protein, which is usually an enzyme, which I have right there. And that is stage 1. So now we have step 2, and the appropriate signaling molecule has binded to the extracellular side of the receptor, which is what is happening right there, and you can recall that from the basic overview of the cellular signaling demonstration I gave at the beginning. And the receptor is activated and changes shape. Its cytoplasmic side binds to the inactive G protein, which is what is happening right here, and the GGP displaces the GDP and activates the G protein, which is the blue right there. So now we are at stage 3, and the activated G protein has dissociated from the receptor and diffused along the membrane and binded to the enzyme, which is now activated. When the enzyme is activated, it can trigger the next step in a pathway leading to the cellular response. And so attached to the G protein is GTP. So now we are in the final step, and the changes in the enzyme and G protein are only temporary because the G protein also functions as a GTPase enzyme. And so now the inactive G protein leaves the enzyme, which is what has happened right here, and it becomes available for reuse. And so the G protein is now available to restart the entire cycle, and that is the end, and then it would go back to step 1. So now the many applications of the G protein are so widespread, you can find it everywhere. It's super important in both your vision and your smell, and the G protein is found in 60% of the medication you find because it's super involved in the bacteria that makes up human diseases. So the G protein is super important and has very widespread functions, including whooping cough, which I tried to demonstrate. So in regards to signal amplification, the elaborate enzyme cascades amplify the cell's response to a signal, and at each catalytic step in the cascades, the number of activated products is much greater than that in the preceding step.